Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Boron, a metalloid element that sits next to carbon in the periodic table, has many traits that make it potentially useful as a drug component. Nonetheless, only 5 FDA-approved drugs contain boron, largely because molecules that contain boron are unstable in the presence of molecular oxygen. It's an entity that medicinal chemists can add to compounds they're interested in, to provide desirable attributes that no other molecule will have, says Ron Rains, the Firmenich Professor of Chemistry at MIT and the senior author of the new study. To demonstrate the potential of this approach, Rains and his colleagues showed that they could improve the protein binding strength of a drug that is used to treat diseases caused by the misfolding of a protein called transthyretin. It contains one fewer electron than carbon and is hungry for additional electrons. When boron is incorporated into a potential drug compound, that hunger for electrons often leads it to interact with an oxygen molecule O2 or another reactive form of oxygen, which can destroy the compound. The boron-containing drug bortezomib, which prevents cells from being able to break down used proteins, is an effective cancer chemotherapy agent. However, the drug is unstable and is destroyed. Previous research has shown that the stability of boron-containing compounds can be increased by appending benzene, a 6-carbon ring. They found that this molecule bound to HIV protease much more tightly than the original version of darunavir. However, later studies revealed that the molecule still did not survive for long under physiological conditions. In the new paper, the researchers decided to use a chemical group called a carboxylate to further anchor boron within a molecule. An oxygen atom in the carboxylate forms a strong covalent bond, a type of bond that involves sharing pairs of electrons between atoms, with boron. This protein, which carries hormones through the bloodstream, can cause amyloid diseases when it misfolds and clumps. Drugs that bind to transthyretin can stabilize it and prevent it from clumping. The research team showed that adding benzoxyborolone to an existing drug helped it to bind strongly with transthyretin. His lab is now working on a new version of darunavir that incorporates benzoxyborolone. They recently developed a way to synthesize this compound and are now in the process of measuring how strongly it binds to HIV protease. MIT has filed for a patent on the use of benzoxyborolone in medicinal chemistry and other areas. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.